This is the second video of recombinant DNA technology or genetic engineering. In this video, we will discuss about plasmid vectors. Now, what are plasmids? These are extra chromosomal DNA present in the bacterial cell. This is the DNA of bacteria and this is the plasmid. These plasmids, they were discovered by scientist William Hayes and Joseph Lederberg in 1952. Now these plasmids basically they are double stranded. They are made up of two strands of DNA like this. So they are closed, circular, and these DNA they are extra chromosomal. This is the bacterial DNA and this is the circular DNA, extra chromosomal DNA, which naturally occurs in bacteria, and this has no correlation with this genome of the bacterial cell. Now this bacterial DNA is independent of this plasmid. Now plasmid can replicate, it has its own point for replication, and this blue point I have indicated, this is its origin of replication. So one plasmid can form, two plasmids, three plasmid, four plasmids. So many copies of plasmid can be produced. So this is called as autonomous replication. So this ORI point, which is also called as origin of replication, which leads to independent replication. Now this replication of the plasmid in the bacterial cell, this can be of two types. A single copy number plasmid, this is called as stringent replication. Multi-copy plasmid, this is called as relaxed replication. Then each plasmid also has a marker gene. Like if we see this is the plasmid, this will have a point which mark itself like antibiotic resistant gene, nitrogen fixation gene. So this is called as a marker gene. It can be for tumor formation. Then each plasmid also has sites for restriction enzymes. What does it mean? Supposing this is the plasmid. Now this will have sites where restriction enzymes can cut. Now what are restriction enzymes? These are the enzymes which can cut at specific places. So each plasmid will have ORI point, which is called as origin of replication. Then this will have a marker gene which will mark itself, like this can be for nitrogen fixation, this can be antibiotic resistant gene, this can be any other for the production of toxic substances, this may be for the tumor formation. So every plasmid may have marker gene. This may have sites for the activity of the restriction enzyme which will cut at specific places. Then ORI point, which is called as origin of replication, which leads to its independent replication. So I just told you that stringent and relaxed replication. So we can just define it. What is stringent replication? Such plasmids, they will replicate only when bacterial chromosome replicate. Like when bacterial chromosome is replicating, mean when this genome is forming two DNA, then only this plasmid will replicate. This is called as stringent. But when this plasmid replicate independent of this bacterial genome, this is called as relaxed kind of replication. So relaxed such plasmids replicates independently of the bacterial genome or bacterial chromosomes. Now these plasmids, which are extra chromosomal DNA having ORI point, that is origin of replication, marker gene, as well as sites for multiple restriction enzymes, these are categorized into different categories. Now we'll discuss about types of plasmids. Now plasmids, depending upon what character they have, they are categorized into different types. Like first we'll discuss about F plasmids. These are the plasmids which carry the fertility factor. So such plasmids, they are called as F plasmids, and these plasmids, they are found in Escherichia coli bacteria. Now these plasmids which have F factor, like I have shown, this is the F factor, this is the ORI point, and this is the bacterial genome. Now such plasmids which have F factor, in these bacteria there is formation of sex pili. These are hair-like structures made up of protein pillin called as sex pili, and they help in process of bacterial reproduction called as conjugation. Now we can see here, this is the bacteria which is not having plasmid with F factor. So this is F minus and this is F positive. So a bridge is being produced. This is called as conjugation canal or conjugation bridge between the two bacteria. So bacteria with sex pili, this can form conjugation bridge with this bacteria. Now this plasmid will replicate and this will come over here, right? So if we see here, after conjugation, this plasmid will come over here. Now this bacteria has become with plasmid with F factor. So this has become F positive, right? So these bacteria, they will get separated from each other after getting the plasmid, right? Now they are called as X conjugates because they have exchanged the genetic material. So sometime this F plasmid, this may get integrated with the host bacterial chromosome. Then it is called as HFR, that is high frequency recombination strain. So such plasmids which have fertility factor or sex factor, they are called as F plasmids. Second type of plasmids are called as R plasmids. Now these plasmids, this is ORI point, that is origin of replication, and they will have gene for antibiotic resistance, maybe ampicillin, maybe tetracycline, maybe erythromycin. So this gene is called as antibiotic resistant gene. So such bacteria which have this plasmid with antibiotic resistant gene, they can grow in that particular antibiotic to whom this plasmid is resistant. Supposing this plasmid is carrying tetracycline resistant gene. Now bacteria can grow in presence of tetracycline. If it is ampicillin resistant gene, then bacteria with such plasmids will grow in presence of ampicillin. So this property helps in gene cloning, as well as this also helps in selection of the bacterial population with the transformed, or you can say manipulated genetic material. So these are our plasmids. Now next plasmids are bacteriogenic plasmids. Now these plasmids, they carry genes for the production of bacteriocins and toxic proteins which can kill other bacteria. Bacteria are sensitive who have this toxic producing protein. They can kill other bacteria. So called plasmids, they produce colicins which kill sensitive Escherichia coli bacterial cell. So they can kill other bacteria. So these are called as bacteriogenic plasmids. Now next is episomes. Sometime plasmid may get integrated. Supposing this is the bacteria and this is its bacterial genome, right? And this is the plasmid, right? Sometime this may get integrated with the just wait. Again make. Now this may get integrated with this bacterial genome. Now when this get integrated with the bacterial genome, then this is called as episome. This may have certain same sequences. This can get integrated with the bacterial genome. This is called as episome. Sometime plasmids, they are quite big in size. Now when they are big in size as compared to other plasmids, quite big in size, so they are called as megaplasmids. Example is Mycobacterium genitalium. Now some plasmids which carry genes for their transfer to other bacterial cell, they help in conjugation process. So they are called as conjugative plasmids. Next category is dissimulation plasmids. Now plasmids which carry genes that form enzyme for catalyzing or degradation of unusual sugars and hydrocarbons. So such bacteria having these plasmids can grow in presence of these sugars and hydrocarbon because these plasmids help in metabolizing them. So such are called as dissimulation plasmids. Now these plasmids which we have discussed, they have a lot of unnecessary sequence which is not required. We are talking about what? We are talking about genetic engineering, right? We want to add our gene into the plasmid. Supposing this is plasmid and we want to insert a foreign DNA into this, right? So we are only interested in this for genetic engineering, for manipulation of this genetic material. This unnecessary sequence is not required. So this unnecessary sequence is not useful for genetic engineering. So scientists, they have designed certain artificial vectors or plasmids for genetic engineering. So vectors designed for genetic engineering. One example we'll discuss, that is PBR 322. P is for plasmid factor, B is for Bolivar, R is for Rodriguez. PBR 322, we can study its genetic map like this. Now this is PBR 322. Now this has this module for replication. This is again a relaxed module of replication mean. This can independently replicate, and this replication module is taken from bacteria Escherichia coli. So this is called as col E1, right? And now this is the origin of replication point, which is leading to its independent replication. And this point, this gene, this is carrying the ampicillin resistant gene. So this can grow in presence of ampicillin. So this is antibiotic resistant gene. Now we can see here, this is another gene which is called as tetracycline resistant gene. So this bacteria with such kind of plasmid will grow in presence of ampicillin as well as tetracycline. Now these are PVU1, PST1, BAMH1, SAL1, HIND HIN2. Now these are the restriction enzymes. Restriction enzymes are enzymes which can cut DNA at highly specific places, right? So these are the areas which can be cut with the help of these enzymes, and we can integrate our foreign DNA, for which we are interested. It's manipulation of the genetic material. This whole genome is about 4362 base pairs in length. So this is the genetic map of PBR 322. ORI point, this point is ampicillin resistant gene, this is tetracycline resistant gene, and these are the point where restriction enzyme connect. So this has multiple restriction sites. So this is artificially constructed vector. This is called as PBR 322. So here I have written the abbreviations. Call E1 is for replication module. So this is showing relaxed mean. This is replicating independent of the bacterial genome, and these are the marker genes or antibiotic resistant gene present in the this artificial constructed vector, and it also has sites for the multiple sites for the restriction enzyme. So this is about PBR 322. So these are different kind of plasmids which we have discussed. Normally occurring plasmids and which are artificially constructed plasmids for genetic engineering. So this is all about plasmid used as vectors for genetic engineering. For how plasmid is being used, like if we make a plasmid like this, right? This has a point which is called as ORI point, that is origin of replication. Now this plasmid is cut from these points. When this is cut, and we can insert DNA into this, then this is joined with the help of various enzymes. So this DNA which we have added into the plasmid, this can be multiplied or cloned, mean its identical multiple copies can be produced. This we'll discuss in the next video in detail. First we'll complete all the vectors. So we have just in this video we have discussed plasmid as vectors and artificially constructed vectors. Now other vectors like phages, cosmids, phasmids, YAC vectors, that we'll discuss in the next video. Thank you for watching my video. If you like my video, please like, share and subscribe.